So we are starting off with this webinar right now. Okay. So I would like to talk in the next 30 minutes about computational biology and its applications as I've understood it in the last few years of my learning and teaching. So it all started off with Gregor Johann Mendel. I believe all of you would have heard of him as the father of modern genetics, and he was the one who actually started off his study by observing. So genetics actually started off as an observational, or biology started as an observational and experimental science, and he went on to do studies with his pea plants. The point was, all of us, I believe, we know about the various Mendel's laws and Mendel's ratios. But the point is that he really did not work with 3 and 4 plants. He actually worked with 20,000 plants. So much of hard work was put in, and out of that, he got ratios and he did a lot of observation. So it was the science was started off as an observational science. There have been a lot of shift in paradigms. I believe you know what paradigm is, right? So it is actually the change in viewpoint. So we've had a lot of viewpoints change in biology and medicine, and right now we are now in an era where we have got so much of technology, and biology has moved on from an observational science to a predictive science. And right now we look into an era where we have a lot of high-end computational organizations which are into healthcare. And I believe you would have noticed when the US president was here, he was also trumpeting about healthcare and how much of AI is playing a very important role in healthcare. So I would like to now probably rush through a few slides, and I would like to give you an overview. This is actually an overview and the basics only about computational biology. So we have to understand that computer is now equally a tool for research, just as a test tube was many years back. And across the years, there have been a lot of people with a lot of landmark events which have happened, which have really brought this science to this area. Linus Pauling was one of those pioneers who actually is considered to be the father of molecular genetics, father of molecular biology. What he was doing was he was actually working on the hemoglobin molecule. And he was one person who told that whenever there is a change in a single nucleotide, when a T becomes A, the glutamic acid, when an A becomes a T there, you find that there is actually a missense mutation leading to a change in the amino acid from glutamic acid to valine, which actually changes the structure of the hemoglobin molecule from its biconcave structure to a sickle-shaped structure. So when he told this was actually the first instance where a molecular conformational change led on to a disease. That was actually the birth of molecular biology. Now, across the years, you would find molecular biology and genetics have played a very important role. And as you see here now, we see a lot of importance given to manipulation of genes, from Watson and Crick's deciphering of the double helix model of the DNA, and then you had gene cloning brought about, and then genome sequencing went on. So a lot of gene-based, genomic-based research has been going on in the sciences of biology and medicine. And this was in 2000, and this was actually after about 15 years of work by 15 countries in the world. And we understand that this is actually President Bill Clinton actually hailing the Human Genome Project as one of the greatest events, even greater than man landing on the moon. And this was actually the Human Genome Project. He was trying to actually announce the completion of the rough draft of the Human Genome Project. So the point was this: after this, the question in everyone's mind was, so what? So finally, at last, after a lot of research, you have actually gotten the complete sequence of the human DNA, human genome, all the genes put together, along with lots of other organisms also. So ultimately, so what are we trying to do now? Where are we going now? So computational biology really came into limelight at this point, but we need to understand that there was already a lot of work which had gone on in computational biology from 60s and 70s onwards. So all these names which you see here, David Lippman, Bill Pearson, and Stephen Altshull, so all of these people were already working in the 1980s itself, where they were trying to look into protein sequences. They initially started with proteins, and then they moved on to DNA, and then they went on to understand how go on to align sequences, and how we try to actually find out or compare, how do you actually find biological insights by comparing protein and DNA sequences. So these are actually, so this gradually led on to 90s, when we started working with the genes, the genome, and the genomic research actually started with the National Human Genome Research Institute, which was actually started, which was the first leadership was under by James Watson, who was actually one of the co-discoverers of the DNA double helix. So it went on into the genome sequencing era, and finally genomes were sequenced, they were assembled, and that is considered to be the largest jigsaw puzzle ever assembled by humans. And later on, we went on to annotate the gene, mark boundaries, where are the genes, where are the introns, exons. So all of those things we went on to understand. Now, later on, this was initially the era started off with candidate genes. When you say breast cancer, we went on to say it was BRCA gene. So it was actually a candidate gene approach which we had. But then finally, when we came to this point where we had to actually, we had technology where we could look at not one gene, but hundreds and thousands of genes put together. So we had this what called high throughput. So you had a lot of output out of exercises like the genome sequence exercises, and that is when we really had to look into computer science, and we need to look into elsewhere for help, and biologists could not just be benchside biologists and do all the work. And later on, it also led on into computational mathematical modeling. Lots of work by allied fields came inside, and we understood that we really need to work as a team. So that's exactly what we are now trying to talk about here. So in computational biology, it is not that we need to learn everything on our own, but we have to understand that things work only as a team. So we had, right now, we're currently in the era of the second and the third generation of sequencing, and we call it next generation sequencing, and lots of transcriptomics. We're trying to talk about genomics, transcriptomics, proteomics, metabolomics, interactomics. So all of these things are actually, we're trying to get what we call, we're actually trying to get the big picture of what is really happening in physiology and in pathology, in normal functioning as well as in disease. If you would like to have a very concise and a very fast moving understanding of what went on in the history of computational biology, you should definitely look into this book by Hallam Stevens called Life Out of Sequence, a data-driven history of bioinformatics. So we had to resort to this. We had to resort to computational thinking. I believe all of those people who initially start off with computer science, actually they learn these basics. It's about when you look into a problem, we try to decompose the problem, looking for patterns, and how do you abstract data, and how do you look for step-by-step solutions by algorithm design. So these are actually things the computer science student studies, but that is what is really required out here when you try to make sense of the sequence also. I would like to first introduce you to a small problem, and I would like to tell you how it is so important for us to look at biology or a biological problem from the eyes of a computer scientist. Okay, so it's a very different way, and it's a very rigorous approach. We as biologists, I actually started my life as a biologist, but then as things went on, when I tried to come inside and see how computational approaches are used here, it was really stunning because it is so rigorous. We don't have such kind of approaches as a biologist, and this is actually, we're not talking about a problem where we're trying to identify in the genome where DNA replication starts. All of us know that cell divides, and before that, it actually has to replicate its genome. And this replication starts at this site called the origin of replication, or replication origin, or ORIC. That's exactly the place in the genome where the gene replication starts, the replication of the DNA starts. Now, when you look into a biologist approach to this problem, we understand that we try to use this knockout approach. We try to remove fragments of DNA, and we try to tell that, okay, if this fragment is missing, what function is missing? So that's actually the exact approach which a biologist talks about. But a computational biologist does not understand this. How will actually a computer recognize the point of origin in a genome on its own? So this is exactly not the way a computational scientist approaches the problem. Now, how does So for example, here we're now trying to talk about a problem where we have actually the genome of Vibrio cholerae. We have here about 500 nucleotides, and here the problem is that where do you find it? How do I know? How do I tell the computer, and how does the computer going to recognize where the replication origin is? So here actually this is the problem of a hidden message. Okay, so when we try to look into this, we already have it's when you write a simple algorithm, it goes like this. You give an input as a string. String means it's supposed to be a short sequence of DNA, and the output is supposed to be the hidden message in the problem. Now, when we try to look at this algorithm, it is not complete. Okay, so when, for example, this is actually a page from a novel written by it's actually a short story by Edgar Allan Poe. It's called The Gold Bug, where pirates have actually gone on to left a secret message. And when you're going to understand what is there in the message, you're going to have actually the treasure. That's exactly what we see in the DNA also. For example, in Edgar Allan Poe's story, you find the semicolon 48 is so frequent. Okay, what does frequent symbols mean? What is the meaning of frequency in a complex problem? That's exactly what we're now trying to solve in the replication origin problem. Now here, for example, this is supposed to be in English. We're supposed to translate this symbolic code into English. So here what we're trying to say is that we are now trying to look into a word which is probably occurring frequently in English, in a paragraph in English. So you find that probably if you look at a normal paragraph, you find the is normally supposed to be the most frequent English word. So you solve this problem of, for example, as semicolon 48 as the. Okay. Similarly here, for example, in the problem of replication origin, also we are now trying to find out a hidden message. And here, when you see, you find in a lot of DNA sequences, you find biological signals. You find repetition of a few nucleotides. For example, here you find AATTT is a surprisingly frequent fimer. Fimer is actually a nucleotide. We understand that oligomer or oligonucleotide means few, right? So fimer is actually a repetition of a 5 nucleotides region, which occurs very frequently in a specific DNA sequence. So we are now trying to actually look at a hidden message, and we're also trying to look at how frequent is that hidden message. So we're not talking about a chimer, we're talking about a fimer, and how frequent is that fimer out here in a DNA sequence. Now, this is actually a little better definition. So when you try to actually put in a problem to the computer, we need to actually be very clear about what is the pattern and how many times of frequency does this pattern occur in a DNA sequence. So this is how we look at it. So this is actually a case of pattern recognition, but this is actually fundamental for us to understand, so that we will be able to educate the computer, or we'll be able to tell the computer in terms of programs to find exactly what we are looking for. So as far as the biology goes, we understand that the DNA polymerase enzyme which performs replication, and it goes on to bind a protein called DNA A protein to a specific DNA sequence called DNA A box. Okay, so when we're talking about a protein DNA binding. So this is actually what happens in biology, but overall, when you look into the gene sequence, we understand that there's a lot of importance given to the finding of these patterns and the frequency of these patterns. So this is actually a simple algorithm which was written here. And here, for example, we are now looking into a 9-mer. Okay, so what is the statistical significance of finding a 9-mer among 500 nucleotides? It's very, very high. So when you find a 9-mer, for example, here it is ATGAT, CAAG, and you also have a reverse complement of that. CATT, GAT, CAT is actually a reverse complement of that. So this is actually a very interesting finding. So we try to actually find a hidden message in replication origin. So these are actually the surprising understanding we receive when we try to look as biologists with a computational problem. For example, in the same problem we are in right now, we would actually, if you look into that DNA sequence, you would actually find 3 mers, 4 mers, 5 mers, 6 mers, 7 mers, 8 mers, and 9 mers, which are also reverse complement of each other. So this is a very interesting find which comes into us as biologists when we try to look into a problem from a computational perspective. So computational thinking and computational biology brings a lot of understanding to life, and it makes a lot of we try to look into concepts with a lot of rigorous, and it makes a lot of hypothesis testing possible. So here in this on the screen, you would be seeing the different algorithms which are used, and the kind of problems which we encounter in biological systems: mapping of DNA, sequencing of DNA, comparing of sequences, exhaustive search, greedy algorithms, dynamic programming algorithm. So all of these are actually the fundamental core. These are actually the core of computational biology. Okay, so it is not totally restricted to this, but algorithms and computational approach to understanding a biological problem is actually is the core of computational biology. When we look into the actual fields, when you look into terminology, you would be finding a lot of things, like you would be finding computational biology, bioinformatics, systems biology. All of these are actually used interchangeably, but there is a lot of meaning to each one of them. And I would be trying to shed a little light on all of this too. So systems biology is the recent one, but we've been looking into computational biology, bioinformatics across the years, genomics. So all of these have been going on. So now, there's such a hype. There is such a hype after the genome sequencing. So we have been looking into a lot of projects which have come about after the Human Genome Project. But then why? Why sequencing the Human Genome Project, sequencing the Human Genome, failed to produce big breakthroughs in disease? Even now we're struggling with cancer, we're struggling with HIV/AIDS. Why is that so? Because biological systems are very complex. Causation of a disease is very, very complex. And we have to also understand that human genome was not really entirely completed. Even now, we do have a lot of lacunae in the way in which sequencing was done and how things have gone about. But there have been a lot of projects which have been started in the post-genomic era, like the HAPMA project, the genome-wide association study, where we've been trying to look at the pathogenesis of complex disease in a deeper manner. This is again another project which was actually carried out by the National Human Genome Research Institute and the European Bioinformatics Institute. It's called the Genome-wide Association Study, where we're trying to understand that there are a lot of SNPs across the genome, and when they are found in association with one another, they are responsible for disease. They're not only responsible for disease, they're responsible for normal functions, as well as they're responsible for a lot of behavioral traits. For example, even suicide attempts are actually mapped to these GWAS, and all of these GWAS studies are done with lakhs of people across different ancestry around the world. So we started off with genomics, it went on to proteomics, and now we are in the era of systems biology, where we're trying to actually get a holistic picture. So what we got from the genome project was actually a parts list. Imagine a lorry. A lorry has got probably thousands of parts. But the point we need to understand is that the parts have a different property than the system. The system, if the lorry is a system which is used for transportation, the lorry's function is totally different from the parts. That's exactly what we're trying to understand here. We were trying to use a reductionist approach. We try to actually bring down the whole organism to the level of the cell, and to the level of a protein, to the level of a DNA molecule, to the level of a single nucleotide polymorphism, a mutation. But then we understand here that it does not really give us a solution. We now have to put together the whole thing, and we need to look into the big picture in order to understand physiology and pathology. Sorry. That's exactly what we are trying to do. So the whole becomes not merely more, but very different from the sum of its parts. So this is exactly what we've been doing. We've been trying to actually touch the elephant, and blind men who are actually touching the elephant different parts, and we're trying to actually understand, but this is not giving solution to the problem. So we now are in search of a holistic understanding of biological systems. So we're now trying to put in, so systems biology deals with RNA, DNA, RNA, cells, proteins, how metabolic networks are formed, and how all of these connect with one another in the big picture. That's exactly what systems biology is all about. So this is actually an interesting study which was actually done not by biologists, but by statisticians sitting out there, and they were trying to actually put together genes, proteins, RNA, and how they interact with one another. The bottom rectangle shows the nucleus, and the top rectangle shows the cytoplasm. How are proteins and genes connected with one another? I'm sorry, this picture is not very clear. So this is exactly what we are trying to do in systems biology. We use a lot of understanding from literature as well as from experimental studies to put together how proteins are localized in the cytoplasm and how they interact with one another overall. This is actually another nice picture which actually shows how diseases are connected. Rheumatoid arthritis, type 1 diabetes, type 2 diabetes, celiac disease, multiple sclerosis, systemic lupus erythematosus. You have a lot of connections. So all of these are very interesting examples where to understand that the understanding the big picture is very important. We saw that Linus Pauling was working on sickle cell anemia. This is sickle cell anemia at another level. So when you try to actually understand sickle cell anemia, especially from the therapeutic point of view, we understand that there are a lot of complex entities which are involved. For example, here we're trying to talk about a protein-protein interaction network where there are specific genes which actually are called the hub proteins. So they play a central role in controlling. So all of the size of the node, we call it, bringing in graph theory from mathematical biology into biology now. So these are nodes and the edges which connect these nodes. So all of these are actually connected with one another, and the large size of nodes denote the role it plays. The larger the size of the node, the larger, the more important the role. So there are a few genes which are actually very important as far because there are lots of genes, but some of them play a very critical role in sickle cell anemia. There is one more allied field to systems biology, that is metabolic engineering, where we try to actually put in a specific metabolic pathway. It could be a small reaction in terms of matrices. Imagine a matrix. This is actually a 3x7 matrix. But imagine a matrix with 20,000 and 20,000. 20,000 cross 22,000. That's exactly what happens in E. coli when you're trying to look into all the reactants and reactions which work in a single cell. So we do have a lot of tools available online. So all of this I've taken from one single article. I'll tell you about this article at the end of this presentation. So we do have a lot of connection between human disease and genes, proteins, etc. So we do have a lot of we do have an understanding about genes as biomarkers. So genes, molecular networks, chemical compounds which work with proteins, how proteins interact with one another, just like genes interact with one another. How do signaling networks connect with one another? So we have a lot of tools for visualizing as well as to understanding signaling networks, metabolic networks. All of these are actually tools, and all of these are links to the tools. I'll give you a link to this article at the end of this. How do you actually go about? So one of the biggest applications of computational biology is drug design. So how do you go about doing it? What are the tools? All of these tools have beautiful tutorials which you can actually look into. Now I'm trying to actually come into the next part of the study where we're trying to look into applications. So this is actually a very interesting study where they used only sequence-based analysis. Only sequences were used, and this was actually a case of how computational biology is used in forensic science. So this is actually a case where you had a doctor, a dentist, who was actually infecting his patients with a needle which is contaminated with HIV virus. Okay, so it was a very complex case. It was actually many years back this was done. I think it was probably done in the 1990s. But then this was actually a case where we understood that just by looking into DNA sequences, we can actually cluster organisms. And HIV was one organism where you have a lot of rapid mutation going on, but then we understand that still you can actually trace the source of origin. Just like we see corona's origin right now, that is one of the biggest problems right now to understand the dynamics of the epidemiology, dynamics of the epidemic. Here, this was actually the first case where where a phylogenetic analysis, meaning a reconstruction of an evolutionary tree, was used for the first time in a US court of law to convict a doctor because he was actually found guilty. Very interesting case of sequence analysis and how DNA sequence analysis can actually help understand HIV evolution and HIV clustering. And right now we are in the in a scenario where so much of difficulty is there. just like we've been having difficulty in finding out an HIV vaccine, you find that coronavirus, though it was a virus which I studied 20 years back when I was doing the microbiology, even now we know there are six strains, and there's one new strain which has come out right now. But the genome sequence has given us a lot of interesting information from which we could actually go on to understand proteins and design vaccines. So this is found on this website called the Protein Data Bank, and you could probably check out, and a lot of information about what sort of structural proteins, non-structural proteins are there in the virus, and how do you go about designing a drug or target the virus with a vaccine, etc. So now we moved on from DNA sequencing, and we moved on into the era where we studied proteins, and we moved on to systems biology. So ultimately, where are we moving on? We are trying to actually look into computational methods where we could actually we're trying to actually model, we're trying to actually look into an era where we're trying to design virtual cells. There was actually a project which is done by the Japanese. It is called the E-cell project, where they were trying to look into, it was supposed to be an in silico cell. They were trying to actually make experiments where they could actually make a software which would actually depict a cell, and you would actually know how genes, proteins, and metabolic pathways work in a cell. And what is the use of that? We could do real-time manipulations. You could actually do a real-time manipulation of all of that, and by that we will be able to understand how cells behave in normalcy as well as in disease. And very importantly, in the last few years especially, we have been generating a lot of data. So why should computer science come into biology? Why is it so important? That's because we have a lot of data to analyze. We don't have tools and softwares. We don't have algorithms to do it. We do not have a methodology. We do not even understand how much of data is there. So much of uncurated data is being dumped every day in public repositories. And here in this slide, you find that we used to call astronomical as a very big measurement of distance and time, etc. But now here and astronomy is also one of the fields which generates a lot of big data. But here you find that genomical is set to eclipse astronomical. Yes, very soon we do have so much of data where even astronomical data by the Hubble telescope, etc., is going to actually be surpassed by the amount of genomic data which we are generating. So we do have a lot of data, even especially with the COVID-19. I would encourage all of you who are taking part in this webinar to go into this website called Kaggle. Kaggle is basically a repository for machine learning data sets, and we have a lot of data sets for the novel coronavirus 2019, and also for the previous episodes of respiratory viruses like the SARS and the MERS, etc. And there have been a lot of competitions which people have been encouraging, the US government has been encouraging, and to go on to forecast the epidemic and the spread of this virus worldwide. And this is actually, this is one of the projects which is found on Kaggle. It is called CORD-19. And this is actually in response to the pandemic, the worldwide epidemic. And they've been trying to actually bring in a team, artificial intelligence specialists from all over the world to look into these data sets to find out what is really happening in the viral evolution. So there's a lot of need for people who would actually look into these kind of data sets. And also, we do have a lot of help, especially, for example, when you look into Data Camp, for example, Data Camp is supposed to be an online portal where you have interactive courses to understand machine learning and what you do with data. And you have a lot of these data sets as well as methodology very specific for understanding how to work with data. And the other very important application of genomics and computational biology is in administration of drugs. It's called pharmacogenomics. Everybody is an individual, and we are totally different from the other individual. And personalized medicine, precision medicine is supposed to be another very important area where, sorry. So we are trying to actually look at an era where biology has become predictive, and medicine is also going to have all of these four components: predictive, preventative, participatory, and personalized. So we've traveled a long distance. We traveled a long distance, but still, we are still far ahead. We're still struggling with cancer. We're still struggling with HIV, and we need to understand that computational biology is so important. It brings an order into our understanding of life. It lets us see the big picture, and it provides us an atlas of life. It gives us a lot of understanding to develop a new hypothesis and test. Have you ever heard of a scenario where a person is called a pipette biologist, or a microscopy biologist, or a cell culture biologist? No, right? It never happens. But when you look at people who are using computers for biology research, their own colleagues do not respect them. They don't really understand. You really got to update. You really got to move out of your comfort zone and to really look into problems, and you could sit in a team. Only then we are going to conquer, because the big problems. This is actually a beautiful slide which shows that cancer is a disease of the genome. And for your kind information, because I believe there are a lot of students out here, even from, I believe there are lots of school students also who are aspiring to know what this is about. There's a lot of future, a lot of money out there. Probably India is really lagging behind out here, but still India is there in the picture. I'm happy about that. But you have a lot of very, very high-end jobs out here in genomic science, in systems biology, systems medicine, as well as in data science. So all of these are companies which are probable prospective recruiters of anybody who would probably specialize in computer science as well as in computational biology. You could be a normal biologist, you could study microbiology as I did, but probably you could just be on the outlook, just open your eyes to the updations which are happening in the field, and that is where everything is important. That is what is everything. That is where the whole field is going on. Just have a look into this quote by Donald Knuth. He was actually a Turing Awardee. It's one of the highest highest awards given in computer science. He says, I can't be as confident about computer science as I can be about biology. Biology easily has about 500 years of exciting problems to work on. It's at that level. We are the ones who give problems to computer scientists. There's a lot of depth in computer science. I do not deny it, but you need to have problems, big data problems, and you need to understand that this could be told about the cell too. Not only is the universe more complicated than you imagine, but it is also more complicated than you can imagine. That's probably so true of the genome as well as the cell. At Sathyabama in the Department of Bioinformatics, we have been trying hard to keep up with the pace where the world is moving, and we have been having a lot of graduates in the engineering sciences with the BTech and MTech degrees in bioinformatics. Right now, we have a BSc and an MSc degree in bioinformatics and data science. So you could probably pass on the message to your friends, and if you're interested, you could very well be in touch with us. So for this presentation, I've referred a couple of very important books, which would also be a starting point for anybody who's interested in computational biology. So this is one of the most beautiful books, and it is a very beautiful book for interactive learning. So it is written by two authors called Pavel Pesner and Philip Compo, and both of them are mathematicians who have actually come in full-fledged into biology and written beautiful books. And both these books are available online. You could purchase them, and they are beautiful books. And in these books, they are actually looking into problems this way. They actually, for every chapter or a unit, they have a question, and they're trying to actually. It is a very biology-friendly book, because if you look into the questions, which DNA patterns play the role of molecular clocks. How do we assemble genomes? How do we sequence antibiotics? How do we compare DNA sequences? Are there fragile regions in the human genome? So all of these actually have a computational understanding which you need to have if you need to actually try to get the depths of computational biology of these problems. Which animal gave us SARS? How did yeast become a winemaker? How do we look at disease-causing mutations? Why have biologists still not developed an HIV vaccine? So these are very, very pertinent problems which probably we need to actually work on. And I told you there's one more beautiful reference which I was going through. I think most of those slides where actually I was showing you lots of links, and with links and tools. So this is actually from this article. It is actually an 80-page review article. It is called Structure and Dynamics of Molecular Networks, a Novel Paradigm of Drug Discovery, a comprehensive review. It is written by this amazing person. His name is Peter Shermally. I hope I'm pronouncing it correct. It is actually on the Web of Science top journals for 2 successive seasons as top 1% paper. This is actually a journal which was published in 2013. And when I was preparing for this delivery, I was trying to look at any better journal and resource for this. I could not find, because it's still one of the best resources for anybody who wants to enter into computational biology and systems biology. And another interesting fact I actually found about Peter was that he's actually, from 2019, he's a student of theology and priesthood at a Lutheran seminary, the University of Theology at Budapest. He's basically a Hungarian he's basically a biochemist from Hungary, but right now he's moved on to theology. What else would you do other than looking to God when you look at complex networks? He's basically a specialist in complex networks. Garry Kasparov, one of the all-time great chess players, was defeated 2-6. He won 2 matches out of 6 matches against a supercomputer called IBM Deep Blue in the late part of 1990s. And he was not a person who doesn't take defeat graciously. And he went through a lot. And right now he gives us a beautiful word. He says, don't be scared of computers. Don't be scared of intelligent machines. Work with them. Learning to work with machines is so important for us in order to keep up with the developments of this age and to be relevant in science and in research. Thank you for your kind listening. Questions, you can post the questions in